Hey, Hammy here. First video here today on Chapter 3, the chemistry of organic molecules. In other words, those 4 basic biomolecules. So this first video is just kind of an introduction to that. You can see the outline of the chapter here. We're kind of an introduction here today on this video, and then the 4 following videos will work our way through kind of each one of those biomolecules. Just a reminder that organic molecules, when we're talking about organic, now Blanchard will kind of tell you a little bit differently in chemistry class, but for biology class, when we talk organic, they contain carbon and hydrogen. So things like CO2, not organic. Things like CH4, methane, organic. You have to have carbon and hydrogen atoms. There are the 4 biomolecules I just mentioned. In the next couple videos, we'll cover each one in more depth. You got your carbs, lipids, proteins, and nucleic acids. And the functions of these 4 are very diverse. We'll touch on some of them. And then as we go throughout the year in AP Biology, some of these keep coming up again and again in different topics. Okay, when we talk about organic chemistry that's based on carbon, we need to review the structure of carbon just a little bit. If you remember, carbon has a mass of 12 and an atomic number of 6. I'm not sure if I put those in the right spot there, but anyway, so 12 minus 6 gives us 6 protons, 6 neutrons, because the atomic number is 6, and then for neutrons, 12 minus 6 is 6. That also gives us 6 electrons. So we put 2 in the first level, and then 3, 4, 5, 6 in the second level. And remember octet rule, you've got an empty spot here, 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 and here. So it tends to form covalent bonds when we talk about biomolecules. And those 4 covalent bonds readily with the chenops, the nitrogens, the hydrogens, the oxygens, the phosphorus, the sulfurs, those elements that are very common in living things, and with other carbons. So the carbon-to-carbon bond is very stable. And this is where you get your carbon-to-carbon, carbon-to-carbon, and you get these long hydrocarbon chains. They can have single bonds. You have carbon-to-carbon-to-carbon. You can have double bonds represented that way, triple bonds. They're all very stable. You can have ring structures. When we look at glucose, we'll look at C6H12O6 and how it actually forms kind of a ring structure. There we go. They also branch off. You have carbons, and then you have a ring, and then you might have a branch of another ring or another chain. And so you get these very complex molecules because carbon is bonding to itself. Off of those carbon chains, then you can have what we call functional groups attached. These functional groups are important because they determine kind of the chemical reactivity, the polarity. Whatever that carbon molecule, that biomolecule that functional group is attached to, depending on what functional groups, it will determine sometimes the shape. If you're talking a protein, how does it coil up into its final shape? If you're talking some other molecules, how do they interact, for example, with water? If you take off a hydrogen ion, just a carbon with a hydrogen, and you replace it with an OH group, okay, that will change it from ethane into ethanol. So it goes from hydrophobic, not liking water, to hydrophilic, being able to mix with water. Okay, so by adding different functional groups, you can very much change the properties of whatever that carbon skeleton, whatever that base structure of carbon is. This is table 3.1 in your textbooks. I do want you to be familiar with this. These are some common functional groups that you find when we talk in biology class, different biomolecules. The hydroxyl group, I want you to know the group. The structure, in other words, R stands for the remainder. That would be the carbon skeleton. So a hydroxyl group would be an OH group. And what that forms, those make alcohols. Okay, so an OH group would make an alcohol. And then its significance, it makes it polar. okay, a carbonyl, carboxyl or carboxylic acid, an amino, and that'll become important when we talk about proteins. Another protein one would be the sulfohydryl, because these sulfide bonds will form disulfide bonds in proteins for secondary, tertiary structures. And phosphate groups. We get into the nucleic acids, the ATPs, the DNAs, the RNAs, phosphate groups. Also in the nucleotides. Phosphate groups in phospholipids. DNA, RNA. Remember, ATP is adenosine triphosphate. Okay, that bond between the second and third phosphate, important. Phospholipids, which we find in cell membranes and other membrane-bound organelles in the cell. Just kind of a side note, when you're talking structural formulas versus chemical formulas, understand that the structural formula might be different. You can have a chemical formula. For example, down here, this would be C3, H1, 2, 3, 4, 5, 6, H6, O, 1, 2, 3. Okay, so for both glyceraldehyde and dihydroxyacetone are both going to be C3H6O3. Their chemical formulas are going to be the same. But when you look at their structural formulas, you see that those atoms are rearranged into not quite the same molecule. So we call those isomers of each other. So just be aware of that when you see a chemical formula, how it's actually put together can vary, can be different. Also, before we get into kind of the videos on the actual biomolecules, it's important to understand that oftentimes we'll talk monomer and polymer. Okay, mono meaning one, poly meaning many. So the monomers are the small things that are going to join together. So it'd be like a monosaccharide, like a glucose. When you join a glucose to a glucose, and you get a disaccharide, maltose. So the monomer would be one glucose. Okay, the polymer would be a whole bunch of glucoses. And if you keep stringing them together, you can get a big polysaccharide like glycogen, where these things, you might have hundreds of these things bonded together. So the monomer is like the one Lego brick, and the polymer is like when you're done building your Legos, it's your final spaceship that you built or whatever. Okay. Proteins, amino acid would be the monomer. There's 20 amino acids our body uses to build tons of different proteins. Okay, thousands of different proteins. And think of them as like letters in an alphabet. Okay, you have 26 letters. How many words can we make? Lots and lots and lots. So depending on how many and what order you join them together, it makes very different polymers. Lipids, we don't always consider polymers because their subunits are not kind of the repeating pattern of something. Sometimes they have very different kinds of subunits, but we'll take a look at that in the lipids video. Again, just a slide showing our different categories of biomolecules. On the next couple of videos, we'll look at these more in depth. Carbohydrates, the monomer is the monosaccharide, kind of makes sense, and the polymer would be polysaccharide. Now you're going to have specific examples within each category, but these are just broad names. Lipids, your subunits, we don't really call them polymers, but the subunits are for like a triglyceride, like we think of fats, okay, would be a glycerol, and 3 fatty acids would make a triglyceride. Proteins, the amino acids, okay, you have your 20 amino acids that you need. We combine those together to make our polypeptides, which are our bigger proteins. Okay, and finally, fourth one, nucleic acids. We use a nucleotide. Remember, a nucleotide is a phosphate group, a 5-carbon sugar, and then you will have some kind of nitrogenous base attached to that. And that's how we make our DNA, RNA. We can throw ATP in there as well. Two important terms that we need to look at is how do we join those monomers together and how do we break them apart? Okay, there's two different things that we use. To join them together, we're going to use a process called dehydration synthesis, or sometimes referred to as a dehydration reaction. Okay, if you look at the root words dehydrate, if you're dehydrated, what does that mean? You're like not enough water. So if you have two monomers, we're going to dehydrate. We're going to take out a water. So we're going to remove an OH group over here and a hydrogen over here, HOH or H2O. We're going to take out the water, and we're going to use these bonds here, right here, to join those monomers together. Okay, so there are enzymes that will cleave or remove those groups off and pull the two monomers together. So joining monomers, joining them together, we're using dehydration synthesis. Now, to break them apart, you have different enzymes. You do the reverse process, okay, which is called a hydrolysis reaction. And again, look at your root words. Hydro meaning water, lysis, meaning split or to chop. So we're going to split the bond between the two monomers, and we have to fill those bonds with something. So we'll take an OH and an H from the water, and we'll put the OH on the one, and we'll put the hydrogen back on the other one. When we get into specific molecules, we'll look at exactly how that's done. Okay, so this is joining, or oftentimes synthesis would be a fancier word than joining, as dehydration synthesis, whereas hydrolysis then is referred to as degradation, okay, degrading, splitting apart. Maybe a more common term would be splitting, joining and splitting, or the fancy term synthesis or degradation, okay, degrading. And finally, just like I mentioned on the previous slide, in order to do dehydration synthesis and hydrolysis reactions, you need enzymes. We'll talk a little bit more about enzymes in future chapters. Just remember, an enzyme is a catalyst. It speeds up chemical reactions, but it's not consumed or changed. Okay, so it'll help a reaction to occur, and it will leave unharmed, and it helps speed things up faster than they would just happen on their own. All right, that's it for the first video here. Cami out.